Is he here? There he is. Just click on this.
It's right there on the table.
Hi, I'm Dan Sinopoli. I am from Hills-Ballston High School, and they'll be presenting my 3-year study that it's been a long time I've been doing this now, and it was kind of the result of one fateful summer from Hurricane Irene. Oh, it was the result of Hurricane Irene. During Hurricane Irene in 2011, the Au-Sable River discharge was over 46,000 cubic feet per second, and which was 200 times greater than its average summer flow of only 235 cubic feet per second. And this picture here is a house that got washed into the Au-Sable River by a feeder stream called Stiles Brook, and that feeder stream normally has a flow of only around 53 cubic feet per second, but during the hurricane had a discharge of 1,200 cubic feet per second. This is a picture that I took 3 years after Hurricane Irene from a helicopter of this feeder stream Stiles Brook, and there's at least 100 large trees in this debris pile, and there are many other debris piles like this that are much larger, and this whole area of devastation lasts for about a mile in Stiles Brook. While fishing makes up only around 2% of businesses in the Adirondacks, it contributes to 64% of positive influences on business retention and attraction since 1990. And what the Adirondacks are really known for is their brook trout fishing. And historically, brook trout have been seen in almost every body of water in New York, including their islands surrounded by salt water. They're very sensitive to the pH and temperature of their environment. They can survive in water about as warm as 70 degrees Fahrenheit and as acidic as 5 on the pH scale. And they also require a dissolved oxygen concentration of at least 5 milligrams per liter, and it's been documented that 7 milligrams per liter or more is optimal. So the purpose of my first year of research was to determine if the environment in this feeder stream, the east branch of the Au-Sable, could support the current population of brook trout after Irene. So I looked at this feeder stream, Stiles Brook, and I also looked at I had 2 other secondary sites, Madden Brook, which flows into Stiles, and it's a secondary stream, and Kiln Brook, which flowed into Madden, which was a first order stream. And I looked at 9 locations within this vicinity, and I looked at temperature, pH, dissolved oxygen, and substrate quality. And where these 2 feeder streams met, Madden and Kiln, there was a naturally formed pond, and during the first summer of 2013, there was a large school of brook trout that stayed here almost the entire summer due to upland groundwater. And so I took advantage of this by taking many underwater videos looking at behavior, and I also netted this one young of the year up in right at the base of where Kiln enters Madden. This is a sample of one of my videos, and well, one of the things that we noticed a lot was this territorial, social agonistic behavior. And the reason behind this was because brook trout predators, they like to obtain food while expending the least amount of energy possible. So all the large brook trout are competing for this area in the middle of the screen. And if that was like the best lie, that was the prime real estate for these fish, because it was slack water adjacent to the flow. Right here, this is Stiles Brook, and this is actually where Madden Brook would flow in. And this large pile of sand that makes this slack water. And so they like to compete for this area. They'll sit here expending very little energy and waiting for food to flow downstream in Stiles, and they'll swim out again and come back. And this video was also evidence of younger year classes, like age 1, year 1 and year 2. And smaller fish were kind of exiled back here in the Stiles flow because they weren't large enough to compete for the slack water. So overall, adequate or excellent substrate quality was found in some areas of Stiles Brook, as they need gravel or cobble to really effectively spawn. And pH was almost always around 7 or 8, except after heavy rain. And if it wasn't neutral, it was high 6s maybe, just slightly acidic. And typical social and predatory behaviors were exhibited in the underwater videos. Another thing I wanted that we noted was the brook trout weren't going for trout worms, we would try to encourage that. We try to document some feeding behavior. We throw trout worms in, and they never went for them, but they annihilated the night crawlers. So excellent. Stylesbrook was excellent habitat for the current trout population, but the question for the second year was, can we figure out if Stiles is able to support future generations of brook trout? And so, after another round of taking temperatures and recording videos, more of the same underwater behavior was observed, and the water quality had continued to be excellent. But then we also added these two variables: aging via scale analysis and substrate quality within a heavily damaged area of Stylesbrook that I had not looked at previously. This is some of the dissolved oxygen levels that we found. We looked at 3 different sites, and it was all well above optimal. But one thing that we noted was that this red bar that was near a beaver dam was much lower during the summer. And the reason we believe that was the case was because since it was near the beaver dam, the water level, or the water, the turbidity was lower. It was exposed to the summer sunlight much longer. The water temperature in that area was higher compared to the other two, and there was also a higher rate of decomposition of organic matter, which can lead to a decrease in the dissolved oxygen. This was some of the summer and fall temperatures, looking at two data loggers, 1 and 6, which were on opposite ends of the stream to one another. And I had 6 data loggers, but I didn't get to have a full profile of Stylesbrook because one of my data loggers actually got stolen by a couple of beavers. It wasn't fun trying to find that. I tried dismantling a beaver dam to find it, and with no success. So at data logger 6, the temperature in like mid-July, the temperature spiked above the survival threshold for brook trout, 70 degrees Fahrenheit. And so it can be assumed that in the area of Stylesbrook around data logger 6, the brook trout were either all scattered in areas of thermal refuge like up on groundwater, or were just not in this part of the stream at all. So, but after this initial spike, the temperature does take a steady downward trend throughout the rest of the summer and into the fall. And so we looked at scale aging for trying to figure out if there were still brook trout being born after Irene, and in this case, there were. Looking at the scales, the brook trout scales, they're almost like tree rings, where the rings are like an indication of growth. And so since brook trout are cold-blooded, they grow less during the winter, and so the rings, the spaces in between the rings are going to be smaller and more minute. So it's like a 1-year increment. It's like a thicker band of rings. That's how you can tell. And so the scale on the left is of a 1-year-old. The scale on the right is of a 2-year-old. This is again, it's evidence of younger year class presence after Irene. The fact that they're still finding young of the year and age 1 and age 2 fish after Irene is very promising. Another thing that we found was a lot of these were generated scales, and this was kind of nature throwing a curveball at us. When we were trying to examine these scales under the microscope, we found a lot of these were generated scales. And brook trout lose scales all the time just due to natural processes like abrasion. But you can't really use these regenerated scales to age fish, just because they only record the age of the fish after the scale itself has been fully regenerated. So when you look at a regenerated scale from an individual brook trout, it could say this trout is 6 months old, but when you look at an undamaged scale from the same individual, it could say, no, this fish is actually 1-year-old or 2-year-old. And that was just something that we noted. We found a lot of these regenerated scales. Sediment samples that I found in this heavily damaged area of Stylesbrook were revealed to be part of a glacial till layer that was, give or take, 10,000 years old, and the sediment was considered glacial due to its characteristics of being unstratified and comprised of different sized substrates. And it was also semi-consolidated. The reason why I say semi-consolidated was because when it was underwater like this, it was very, very dense. It was like we were walking on concrete or pavement. But then when it was exposed to air like this, in figure 11, it was very brittle and easy to break, mainly due to the fact that it was comprised mostly of clay. And this whole area, due to this density, due to the density of this till layer, it's impossible for brook trout to even spawn in this area, because there's no way brook trout's going to be able to dig into this till to make a red. After year 2, we discovered a history of stocking that dated back to 1929, and ending in 1993 when Styles Brook was last stocked by the Randolph Fish Hatchery. So I had two questions for this last year of research. Is there a genetic difference in today's trout population from the original domestic strain 22 years ago? And are the individuals in Styles genetically similar to trout in an unconnected stream of close proximity? And to do this, I collected 90 adipose fin clips as sources of DNA, 30 fin clips from Styles Brook, 30 fin clips from Spruce Mill Brook, which is actually a feeder stream to the Boquet River, and we took fin clips from trout in an area of Spruce Mill that had been considered to be wild, and 30 fin clips from the Randolph Fish Hatchery, which contained a domestic strain. While these two streams, Styles and Spruce Mill, they're very close to one another, they're on the same mountain, the trout that lived within these streams looked pretty different physically. The Spruce Mill individuals tended to be smaller and darker, and the reason we believe why they were darker was because it was a lot more canopy, and that favored the darker individuals, so they were able to pass on those genetics for the darker color to their offspring. And the Styles individuals tend to be on the larger side. They also were lighter in coloration due to the fact that it was a lack of canopy. In many areas of Styles Brook, it was just total sunlight exposure. All collections were recorded between July and September of 2014, and the only small fish that were caught in the field were recorded. Brook trout were actually caught via angling and were hooked in the lip with small hooks to reduce injury. And as an extra precaution, brook trout were placed in buckets of cold stream water with added ice to kind of lower their metabolism and also numb them before the procedure. Unharmed brook trout were released back into their natural habitat, and fin clips were stored in 95% denatured ethanol until analysis was performed. DNA was isolated using a modified pure gene DNA extraction kit at the USGS Leetown Science Center in West Virginia, and the samples were screened at 13 different microsatellite loci designed for brook trout. And this table down here are all the microsatellites and the number of alleles tested with each microsatellite, and there's a total of 92 alleles tested. I put together this light frequency histogram for all the brook trout that were caught in the field, just to show again all the Spruce Mill fish were indeed on the smaller side, and the Styles fish were all on the larger side. The average Spruce Mill fish was around 13 centimeters, and the average Styles fish was around 16 centimeters. The reason we believe this to also be the case is the ease of accessibility for Styles Brook. Spruce Mill and Styles are on the same mountain, but Styles is more towards the base of the mountain. There are areas where you can literally fish out the window of your car and catch a fish. It's that easy to fish in Styles Brook. So this ease of accessibility leads to the potential for increased fishing pressure, and with increased fishing pressure comes fewer individuals per given area, which with less competition can lead to these 22 and 24 centimeter fish, which were much larger than the average. This is a principal core analysis for proportion of shear alleles distance, and if Randolph and Styles still shared alleles, the ellipsis should be